Welcome to another episode of the Neurobiology podcast with Said and Nika. This is a conversation with Dr. Arezu Jahani-Asl. She's a neurobiologist and Canada researcher at the Brain and Mind Institute, affiliate investigator at Ottawa Hospital Research Institute, and associate professor in the Department of Cellular and Molecular Medicine at University of Ottawa.
During her scientific career, Dr. Jahani has investigated the molecular mechanisms that regulate brain tumor development and progression, with a particular focus on glioblastoma. Her lab studies how signaling pathways and cellular interactions within the tumor microenvironment influence tumor growth, invasion, and resistance to therapy. By examining the roles of oncogenic signaling, stem-like tumor cells, and interactions between cancer cells and surrounding brain tissue, her research aims to uncover vulnerabilities that can be targeted for new therapeutic strategies. We hope you enjoy this episode, and here is Dr. Arezu Jahani-Asl.
All right. Good afternoon, Dr. Jahani-Asl. Welcome to our podcast. It's an honor for us to have a conversation with you. I've been really looking forward to this episode, been preparing the questions. And I remember looking at your website last year, looking for different labs. And it was one of those websites that I spend a lot of time because I just kept reading and okay, there's a lot going on.
Very interesting.
So can you start by telling us a bit about your journey? So where was this story beginning of you becoming yourself to this area of research that you're interested in right now? Educational, different.
So I was born and raised in Iran, and I did my undergrad at the University of Toronto, my master and PhD both here at U Ottawa. Did a fellowship postdoc for 6 years at Harvard Medical School, and I got my first faculty position at McGill University. And I moved to University of Ottawa in 2021. So it's been all medical research, and I've been finding my interest along the way of what I want to focus on. We have different research programs, as you can see from the website. We work on intellectual disability, neurogenesis, stem cell fate specification. But the biggest program in my lab is brain cancer, specifically glioblastoma, and that's what I did my postdoc on. Obviously, there are different components to it, bringing my PhD expertise into it, looking at mitochondria in the context of cancer stem cells and transcriptional regulation, continuing on my postdoc studies. But mainly, we look at cytokine signaling, focusing on a receptor that I worked on for 15 years. So this receptor is called oncostatin M receptor, or OSMR. When I started focusing on this, there was not too many PubMed citation. There was probably a few paper. And I came to focus on this after I did some ChIP-seq analysis, which is chromatin immunoprecipitation, to see what the STAT3 regulate, because our major focus is EGFR-V3 STAT3 signaling. And every time I was analyzing the ChIP-seq, I came to see this huge peak. It's this should be for a reason. It can't be just a fluke. So, and then I had consultation with my advisors and my colleagues, and it's oh my God, you got so many other cool hits. You should focus on those instead. And I thought, no, I'm going to study this receptor. So I started from from scratch, basically, and it got interesting. And I had another project, postdoc, on X-linked intellectual disability. Once this OSMO was picking up, I completely dropped that one. We still had a paper on that, but I continued studying OSMR, and it became more interesting, doing different things. And these days it's really picking up. There are so many. I did a recent literature search. There's so much being discovered about oncostatin M receptor in the context of other cancers, in regulation of immune suppression, in the tumor microenvironment. So it's picking up. it's starting to pick up. And I'm happy that I was one of the first to show this so important glioblastoma as part of EGFR variant 3, which is very difficult to target signaling. So that's the story.
So now let's focus a bit more on the receptor. How put it in the context of glioblastoma? Is it a matter of overexpression? Is it a matter of mutation in terms of misfolding of the receptor?
What is the role of this receptor in terms, like in connection to cancer?
These are good questions. So to answer the first one, this receptor is rarely mutated in the context of cancer. So it's doing what it's doing via signal transduction, signaling, and providing a platform because it's a required co-receptor of other oncogene. And it's really upstream of key transcriptional factors like STAT3 that regulate genome reprogramming during tumor progression. And it's also, in terms of what it's doing, it's operating upstream of PI3 kinase, AKT, MAPK, ERK. So pretty much it's a master regulator, right? It's activated in response to a ligand, oncostatin M, binding the receptor, inducing all these different pathways. The major one we're focusing on is STAT3.
Makes sense. So you said that this can serve as an alternative therapeutic target because we cannot target the epidermal growth factor receptor. How would you target this receptor?
This receptor is much simpler to target. It's still difficult, but less challenging. Because in case of EGFR variant 3, this is RTK, receptor tyrosine kinases. So there are a lot of compensatory pathway. One is called bypass pathway activation. When you knock down or you inhibit EGFR or EGFR V3, PDGFR get activated, does the same thing. EGFR family, the mutational spectrum is very wide. It's very difficult to target all this variant. Whereas this is a one-span type 1 receptor. It's not highly mutated, as I said. And its targeting is specific. And one more important thing, this receptor is not expressed in normal tissues to a big extent. It's highly expressed in cancer tissue. It's expressed in embryonic brain too, but its expression significantly declines after birth. So by targeting it, you're specifically targeting cancer cells without inducing much side effect to non-cancer cells. So specificity.
And you mentioned that it's also something specific in the matrix of the cancer, so in the whole environment. How is can we look at it in the sense of how the cancer cell is communicating with its environment and how it's separating the matrix from healthy cells?
Yeah, one important component is that this pathway operates in a paracrine fashion, where the ligand is mainly secreted from the macrophages or tumor-associated macrophages and binds the receptor, which is highly expressed on the solid tumor tissue cells. So this is the loop that connects the microenvironment to the cancer cell.
And is this receptor also relevant to other cancers other than glioblastoma?
Yes. So as I say, we're first to show in the context of cancer, among first at least as far as I know, in terms of brain tumor. There were a couple of studies before us, I think on colon cancer, but then now it's established another landmark study that came out. It's showing this paracrine signaling was in breast cancer. I think it was a JCI paper, and then another one on pancreatic cancer. I think the pancreatic cancer was published in the journal Gut. These two were really showing exact same thing in a different context, but now it's increasing. I saw it in muscle-associated cancer, muscle atrophy. There are other models that being studied, and the pathway seems to be common. Ovarian cancer,
That's
several,
really fascinating
several
because
malignancies. Yes.
it's really tough to find like all cancers are like
Yeah,
different in all. So
yeah. This is a common pathway, and that's interesting.
So, what are the opportunities for? So, we started asking about targeting this receptor. What are the opportunities and also challenges regarding into designing perhaps pharmaceutical or agent that could?
So that's a major focus of our studies. One is to basically, one focus has been repurposing FDA-approved drug. We have done drug screens, to find small molecules that can inhibit OSM-OSMR signaling. Our readout is being STAT3 phosphorylation. Another is to map key interactors of this OSMR protein that crosstalk. We have a new study coming out hopefully soon, where we have shown that this receptor crosstalk and bind directly to a chloride ion channel. And we have designed peptide to inhibit the interaction so that we can impair signaling. There are many different ways we can identify phosphorylation sites and make site-directed mutagenesis. And all these are great opportunities, especially if you want to get a compound or an inhibitor or a small peptide into the brain.
And what are the main challenges that you have encountered designing these molecules?
Challenges are daily.
Of course.
They never go away. I spent the first 3-4 years of being a PI, making an antibody. We were well funded for it. And unfortunately, it didn't work out at the end due to some conflict. So we had to switch. So this was one big challenge because I fell behind for years. Then I started over and we did a compound screen, and the results are promising. We have a lot of hits being validated right now. So we have our top 10 to go through. And in addition to that, all the molecular mechanisms that we're trying to map, each of these provides new opportunity.
Exactly.
If you find the interactor, inhibit the interaction. If you find the phosphorylation, inhibit the phosphorylation. And if you find how the binding occurs, mode of binding, we do a lot of protein modeling. We can also target that way. So there's many different avenues we're exploring.
And how are these getting into the brain? How are these getting past the
Blood-brain barrier, yeah.
Because that's also
So yes, so it's different approaches in terms of the antibody. It doesn't get into the blood-brain barrier. So in preclinical model, we have a miniaturized pump to get it to the brain. Of course, in clinic for a human patient, there are other approaches like neural stem cells, route of delivery, adenoviruses, or somehow they generate different protocols, which is not our expertise for clinic. But the other compound or the other inhibitors we're focusing, they're mainly peptides. They're very small, they get into the brain. And for the FDA-approved drug, we have limited our focus to small molecules that can pass blood-brain barrier. Just chemistry.
Hardcore chemistry.
And do you see the scope of your research in the future also getting into the other? Because you mentioned that it's not as specific to glioblastoma, so it's also seen in other, which is also something that's very exciting about it. It's also seen in other cancers.
Yeah, it's certainly applicable. Any good drug against this pathway, it can be used for others. It's even simpler because we have the challenge of blood-brain barrier. They don't, right? So then how you get this into therapeutic window, that's the next phase of project. So to kind of figure it out.
All right, now taking a step back, what initially drew you to neuroscience? Why not the other subjects like metabolism, immunology? Why neuroscience?
Or was neuroscience the focus first,
Or like
you
you
just,
changed?
or the other time
Yeah.
it was just because the cancer was in the brain? It grew.
I did my PhD in neuroscience. So, yes, I switched to neuroscience because I was fascinated by the brain. You know, there are always so many questions that we still need to answer. I started my master, I did ovarian cancer. That was also a very deadly cancer. So then for my postdoc, I combined both brain and cancer. Yeah, it was a good match.
And how does the other streams of research that you have in your lab help or crosstalk with this particular focus on the receptor?
You're asking good questions in many different ways. So in terms of biology, our focus is transcription, genetic, epigenetic mechanisms that regulate sulfate. In the context of my other project, we look at sulfate working on neural stem cell. In the context of cancer, we work on cancer stem cell, brain tumor stem cell. So, and then we look at the transition of the mechanism that can turn a neural stem cell into the cancer stem cell, right? So it's very connected, with the general theme being transcriptional regulation. And the other thing is sulfate. Mechanisms that regulate sulfate always overlap, whether it's pro-differentiation program, proliferation, sulfate dormancy. You know, these are all the common theme that's common between the two projects, but in the context of different diseases.
Now that you mentioned dormancy, how do you target that?
So dormancy is a big topic right now, actually. It's been the theme of number of grant theme in recent years, especially when it comes to cancer. It's a big problem because most of the chemotherapy and ionizing radiation, they target dividing cells. So it's always the dormant cells that get away and then re-enter cell cycle and induce tumor recurrence. So we know very little about dormancy. A couple of pathways known, but it's being investigated. So definitely this is an area we need more research.
The problem with the targeting.
Yes.
All right, now switching gears a bit, you also teach beside doing research, correct? So which one do you prefer?
I like both, but I don't have time for both. So if I have to choose, I would choose research. You know, I like human interaction and helping students. I have a son, so I know it could be very impactful, but research is bring you different realm of excitement, you know, and it's that eureka moment, I thought. So I prefer research, but I like teaching too. Although I believe you can be a great researcher and a great teacher. It's you try, but it's like, you know, being a great scientist and being a great mom doesn't work together. So always.
For teaching and research, the way that I think about it is like, the research itself is just being at the edge of figuring out the unknown. But if you want to teach, you have to step back and try to communicate it. So it's like this cycle of going from this one to the other one.
So the challenge of teaching is you don't get everybody in a classroom is not the same or at the same level, or it's also different. You get different personality types. One style doesn't work for all. So that's the challenge for teaching. The other thing is teaching at advanced grad courses is much related to me because I can teach them about my research program. And but when you put like third year TMM, or it has to be really basic, and it's very difficult to find, you know, for a researcher to find that common ground. How do I make this very basic? Sometimes I make it so basic that like, am I gonna teach this?
What do you want your students to remember from the courses or the classes that you teach?
I want them to remember one take-home message related to the lecture. Depends what the lecture is. So if I talk about glioblastoma, I want them to remember what's the present challenges, why these tumors are incurable, why they always recur, what are the recent research that's going on. And I want them to take away one key pathway that they can describe to their friends.
Now, to also just talk about the glioblastoma itself, I don't know that much about, like, is there a particular age group that are mostly affected, or it's not really related, it's
And
just
also,
a matter of genetics?
yeah, and how the grading system works, like, because there's like grade 1 and 2, which are like more less severe, and then we have 5 that is the most severe, if I'm not mistaken.
There are initially 4 grades according to World Health Organization. 4th is deadly, obviously. 3, 4 are very malignant. 3 can turn into 4. 1 and 2 are more manageable. Things are changing. Now we're adding, this was initially based on histology and, characterization of the tissue. Now we're adding into that genetic heterogeneity, mutations associated with grade 4. In terms of age, it mostly happens in adults of 50 plus for adult glioblastoma, sometimes 40 plus. Most common in male than female.
And is there like a specific group that are more prone to it? What is the origin of glioblastoma?
The origin of glioblastoma, in terms of its cell type, it's known to be progenitors or perhaps uncommitted neural stem cell at earlier age that later become oncogenic progenitors. Previously, since glioblastoma is mostly composed of astrocytes were suggested to be the cell of origin, but recent single-cell studies have shown it's mainly the progenitors, and there has been other hypotheses, the differentiation of astrocyte into brain tumor stem cells and so on. But recent research mostly support that the progenitors are the cell of origin.
It's very interesting that you mentioned other than the cancerous cells in glioblastoma and other cancer cells, we find this particular receptor in the embryonic brain as well. So it makes sense in the light of, the embryonic cell is doing a lot of growth. And also that's also what we see in cancer. Now, and the age group that you mentioned, that it's 50 plus. What's interesting to me is that, in that age group, it's also the start of the onset of a lot of other cognitive problems, such as subjective cognitive decline, mild cognitive decline, and then Alzheimer's and dementia and all of that. So there's a lot of effort to cure or treat those conditions as well, which will translate into, now we're going to have a lot of plasticity again in the brain in different areas. Now I wonder, would that shift the balance toward higher risk of cancer?
That's a very interesting way of thinking about it and trying to connecting the two. Actually, I saw a paper last night. I was just doing Bubble search. There was like this neuroplasticity manipulation for glioblastoma treatment. Yeah, there are definitely, and there is some research going on. So there are lots of epidemiology research as well correlating, how many Alzheimer's people get glioblastoma, and I'm not up to date on those statistics, but it's an active area of research.
It's very fascinating. It's just, as you mentioned, the brain, there's a lot of questions that need to be answered. Every single little door turns into 1,000 on its own.
Yeah, he also does research on brain and schizophrenia, so
Yeah. What do you work with?
Dr. Northoff is the Royal Mental Hospital.
Yeah.
So now we can ask about you outside the research,
Yep.
the research area. Do you have any hobbies? Any particular thing that you like, taking walks? Or it's interesting because we asked this from all the other guests, and interesting stuff come up, like cooking. A lot of them enjoy cooking.
Or running.
Yeah.
Definitely, like any other human, I have a lot of interest, but do I have time? No, because I spend a lot of time on my research, and especially when you're postdoc and new PI, you don't have time for anything else. Well, I didn't. Maybe it's just the personality type. Some people do make time for it. But there is and if you have a family and, aging parents to take care of, teenager and all that, you try to make time for them. And then after you make time for everything, there's very little time left for your hobbies. I mean, it's different if you're single or you don't have kids or you don't need to take care of your parents. But for me, it's been very busy. I used to, before starting my postdoc, play music. I play sitar. So I really like that. I like to dance when, there is a party. Yeah. I have a lot of other interests that I've always wanted to do, but maybe I'll do them when I retire.
And there's a time.
Yes. Yeah, those are good to do.
If you weren't a scientist, what career path would you have chosen?
I have had quite a few other options. Actually, I wanted to be an architect, designer. Mm-hmm. I like fashion. So these are areas that I would have focused, which I think it's connected to what I do. I design experiments, which kind of satisfying. I generate figure, and aesthetic is very important. So it still helps satisfy myself, but I really wanted to be a designer. And I go shopping for the fashion.
There's little bits of it here and there.
Yeah, yes. So I get a little bit of all, but I would have been very bored, basically, if I wasn't a scientist, because some brains don't get that satisfaction easily. I mean, feed them with reading a novel, watching news or series, or after 1 week, I could get bored. So, whereas being a scientist, there are always so many questions keep you busy. So you don't have time to get depressed or bored, or, it's always, you keep it going. It keeps you on your feet. Yeah.
Well, and now the big last question. What is the meaning of life?
Wow. And now it's getting philosophical. Yeah. It's a question we all have at different stages of life. Maybe if you asked me this question 20 years ago, I would have responded differently, but with the age come maturities, I don't know. So I think it's different for everyone. Life is what we give it meaning, From my perspective, life is when you're in peace with yourself. You're in peace, you're comfortable with who you are, and you're genuine, you're yourself, you contribute genuinely, you make sure you're well, healthy, mature, and once you're well, you can make life better for other people too. So my advice to trainees is to focus on the quality of their daily life, control what you're thinking. Don't just, when something is giving you a negative feeling, just stop. Don't think about it and turn it into positivity and anything that makes you feel appreciated. And usually young people, they criticize themselves a lot. I could have done better. I should have done this. Just don't. So appreciate little things. enjoy every single thing that's going on around, and if you don't feel good about it, just don't do it. And once you do that, you create a nice life for yourself and for others.
That is a beautiful answer.
Yeah. Thank you very much.
Yeah.
You're welcome.
It was a pleasure having you on the podcast.
Thank you for inviting me.
Thank you very much.
Thank you.
Thanks.
Thank you for listening to this conversation with Dr. Jahani-Asl. We discussed her path to neuroscience, her work on glioblastoma, and more. If you're interested in her work, you can find the link to her website in the description.
This is a Dendrite podcast. To support it, please share this episode with your friends and anyone else who might be interested. We hope you enjoyed this episode and see you next time.